All right, so we're going to finish up this chapter by talking about extracellular components of cells, and then the connections between cells that are going to help to coordinate cellular activity. So because we're extracellular, remember that we're outside the plasma membrane. Most cells are going to synthesize something, they're going to secrete materials out of that plasma membrane. So they're going to push stuff externally outside of the cell. So we have some structures to contend with out there. In plants, we have a cell wall. Remember we said that the cell wall is technically outside the cell because it's on the outside of the plasma membrane. In animal cells, we need to talk about the extracellular matrix, or the ECM, and what that does for us. And then the third thing we're going to talk about are intercellular junctions and how those cells are attached or joined together, and what those different type of junctions will do for us. So let's start with cell walls. Cell walls of plants. So remember, we're talking just about plants. So the cell wall is an extracellular structure. It's one of the distinguishing characteristics between plant cells and animal cells, along with the chloroplast and the central vacuole. We'll see prokaryotes and fungi and some of the protists also have cell walls, so not just plants, some others as well. What does that cell wall do? It protects the plant cell. So it's going to maintain a shape, it's going to prevent excess uptake of water, it's going to be a tough barrier from external or harmful environmental things. It's made up of cellulose. So you have cellulose fibers that are embedded in other polysaccharides and proteins, and it's going to be that tough, grainy, or woody component that you're used to seeing in plants. So that's what it's going to do. And then we've got a slide here on extracellular matrix. We're going to see support, adhesion, movement, and regulation. Not really entirely sure why that ECM is right there. I would have put it on the next slide. So let's look at this image and then we'll grab these ECM functions again. Weird spot for that. We're going to have to go back and talk about that. So plant cell walls have multiple layers. They have a primary cell wall, a secondary cell wall, and a middle lamella. So the primary cell wall is relatively thin, it's flexible. There is a middle lamella, so that lamella is not in between the primary and secondary. It's actually between a primary and a primary of another. So we'll look at that visually in a second. But the secondary cell wall is added between the plasma membrane and the primary cell wall. Okay, so let's take a peek at that. So here's what I've got. I've got one cell, here's another plant cell. Okay, so I have this inner part right here. That's a secondary cell wall. Same thing over here. I have this big, thick secondary cell wall. And then right outside of it, I have a primary cell wall of one cell. Here's the primary cell wall of another cell. And in between the primary cell wall of this cell and the primary cell wall of that cell, I have this part called the middle lamella. Okay. Okay, so you can see that here. So I have this secondary cell wall, I have this primary cell wall, and then I have this gap in between, right? And that's the middle lamella. Something else I want you to notice while we're on this image, because we're going to back up a slide, you see these bridges between the cytoplasm of one cell to the other. See how this is the cytoplasm with the central vacuole and the chloroplast and the mitochondria. See these bridges that cross the cell wall between all of these cells? Those are the plasmodesmata. Plasmodesmata are channels between adjacent plant cells, and they allow for the transport or the transfer of material from one cell to another cell. Okay. So you've got some vocab here. You've got a primary cell wall, a secondary cell wall, a middle lamella. You've got plasmodesmata. As far as cell walls go, you want to know what their function is and what they do. We need to grab this part right here and talk about it in extracellular matrix, which we're going to do right now. So extracellular matrix is something we talk about in animal cells. Animal cells lack cell walls, but they have this very elaborate extracellular matrix. It's made up of glycoproteins like collagen, proteoglycans, fibronectin. So it has this substance in between the cells. that extracellular matrix, or that substance, binds receptor proteins to the plasma membranes. We call those integrins. The other the other thing that they do is that extracellular matrix is going to provide support to the cell, the same way the cell wall provided support in the plants. We're going to see adhesion. So remember from our chapter on water what adhesion means. We're going to allow for movement and then regulation of substances. So grab these four and get these four functions over in your extracellular matrix. Okay, so this is what extracellular matrix looks like. So this is inside the cell. Here's your lipid bilayer that is the plasma membrane. We have these integrins that we'll talk about. And out here is this extracellular matrix, or this extracellular fluid. These big, thick strands with the light and dark purple banding on them, those are collagen fibers. We're going to see some fibronectin that typically will be a bridge or a binding between the integrin or the protein within the plasma membrane and collagen. And then this green vine-like structure that you see floating around here out in this fluid, those are proteoglycan. So protein, glycan, sugar, or carbohydrate. Okay, so they're a protein and a carbohydrate, or a protein and a polysaccharide molecule that moves and weaves in through and back and forth in this space. So that space, we're going to use this space to diffuse across. This is going to be it's going to be spacing between cells. We're going to move substances across these areas. This is going to be the site where we're going to see some receptors and cell signals that are going to be out here that identify these cells. So we'll talk about that a little bit more. But several types of junctions, intercellular junctions. I'm not going to make us learn them all. I'm going to pick the big ones that we need for 181. Recognize that when you get to 201, you're going to add to this list. So this is not a complete list, but you're going to add to this list. But let's grab the big ones that we need for what we need here in 181. So what intercellular junctions do is they allow neighboring cells within a tissue, within an organ system, to adhere to one another, to interact with one another, to communicate through a form of direct physical contact. So that is what an intercellular junction is. Inter is between cells, so it's a junction between two cells. We're going to talk about these four. Again, not a complete list. Plasmodesmata, tight junctions, desmosomes, and gap junctions. So again, here are your plasmodesmata. We looked at those when we looked at the primary and secondary cell walls and the middle lamellae in plants. But plasmodesmata are channels, or bridgings that perforate the cell walls, and they allow for solutes to pass from the cytoplasm of one plant cell to the cytoplasm of another plant cell. Things like water, some of the small solutes, sometimes even some proteins and ribosomal RNA can pass from one cell to another, but that's the job of a plasmodesmata. In animal cells, we talk about tight junctions, we talk about desmosomes and gap junctions. Again, not all of them, but let's get these three down for here. So tight junctions are junctions between two cells that are going to be leak-proof. They're going to be waterproof. They're going to adhere to neighboring cell membranes together, and they're going to prevent leakage of extracellular fluid. Okay. Desmosomes are going to be anchoring junctions. They're going to fasten cells together in tough, strong sheets. And then gap junctions are going to be a form of communicating junction. They're going to provide a channel, kind of like the plasmodesmata did in plants, but they're going to provide a cytoplasmic channel between adjacent cells. The beautiful thing about gap junctions is they can seal off on either end. So if one cell needs to, it can seal itself off from the other cell. Here's an artist's rendition. Okay, so again, not all of them, but just the big three we're looking at. So we can have a tight junction here, right? We can have a tight junction whereby we're going to create a waterproof, leak-proof seal where we're not going to let anything in or out of this space. Okay. We can create a desmosome where we have an anchoring plate in each cell, and you'll see bridgings across there, kind of like snaps or rivets. There's a plate on each side, and then they rivet together. And what you're seeing over here are anchors. So you're seeing anchoring intermediate filaments that are going to anchor this desmosome plate at the cell wall or the cell membrane. They're going to anchor it into the cytoplasm. And then gap junctions are more like the nuclear pores that we looked at. So they're going to be these open channels that will go between cells. Next slides are, actually I'm going to pause here and then I'll just do those last ones because we're slightly over 10 minutes.